take a look at the overall functions of the kidneys. So kidneys, the main structures, the main organs of the urinary system. And the kidneys, you have two of them. They're sitting in what we call the retroperitoneal area, so right behind the parietal peritoneum, right? The peritoneum is connective tissue that covers all your digestive organs and things like that. So you peel that away, you remove all of your digestive organs. What you see is the back of that particular area, where you have like your abdominal aorta, your inferior vena cava, and then you have two kidneys on both sides. Now, the overall functions of the kidneys is to essentially regulate the amount of ions or the concentration of ions inside your blood, okay, inside your plasma. So if you have too much of those ions inside the plasma, the kidney's job is to get rid of it. If you have too little amount of ions inside the plasma, the kidney's job is to take that back from the kidney itself, right? Because essentially, you're gonna have a series of what we call filtration between the blood and the kidneys. So the fluid components of blood is gonna be filtered through the kidneys, and all those contents will be placed inside the kidneys themselves. So the kidneys then have to have the ability to take those substances back so you don't lose them, right? So the type of ions that they regulate will be sodium ions, potassium ions, chloride ions, and anything that you can think of. Now, in addition to ions, the kidneys also help regulate certain organic compounds like glucose and amino acids and things like that. Okay, so the glucose that you have inside your blood, the amount of amino acids that you have inside the plasma will also be filtered into the kidneys themselves. And then as they're circulating through the kidneys will take them back, as necessary, right? Now, as the kidney is regulating all of this ionic composition inside the blood, it is also then acting to maintain what we call the blood osmolality or osmolarity. And of course, we didn't really talk about this term, but you guys know what osmolality is, right? Osmolality is the collective amount of molar ratios of every single solute that exists in that solution itself. So imagine whatever those particular solutes are inside your plasma, you have to add all of those concentrations together to get your overall osmolality, okay? And that's gonna help maintain the third thing, which is what we call blood volume. Okay, because of course, when you have osmolality, it's gonna draw in water, it's gonna maintain some degree of volume inside the plasma itself, right? Plasma volume is gonna equate to about your blood volume. And your blood volume is important because we already talked about the cardiovascular system and how the heart pumps the blood throughout all your blood vessels so that you can deliver adequate amount of blood volume to all your cells and tissues of the body, okay? Now, a fourth function for the kidneys is to essentially regulate a very important type of ions that we have inside the plasma, and that is hydrogen ion. And we all know from our discussions, obviously throughout the whole semester, that hydrogen ions, wherever it exists, is going to impact, or basically it's going to have some kind of effect onto the pH of that solution. So the more hydrogen ions you have, the lower pH you have, the lower amount of hydrogen you have, the higher the pH you have. So therefore, to regulate the pH of the blood, the kidneys will act to regulate the hydrogen ion concentrations as well, okay, to maintain normal blood pH. So when it comes to pH balancing in the body itself, and especially in the blood, there's two main organ systems that will help you do that. One is the respiratory system to help regulate the amount of CO2 that you have. Two is the urinary system to regulate the amount of hydrogen ions and bicarb that you have. Okay, so it's all very important for the pH of the blood. Now, of course, there's a lot of substances that are metabolized throughout the whole body, right? The cells are breaking down constantly a bunch of these particular carbon sources, so they're constantly making waste products or metabolic waste. One of the areas that you can get rid of this metabolic waste is through the urine. And the structure, the organ that is actually responsible for producing that urine is your kidneys. So therefore, some of your metabolic waste throughout the whole body that is being generated by all these cells will be excreted by the kidneys themselves. Okay, so you have to get rid of these metabolic waste in some way, shape, or form. So you can imagine if your kidney starts to fail for any particular reason, let's say if it gets poisoned by some kind of toxicity, or because it has failed because of some underlying medical condition or whatever the case is, then all of these functions become affected. If you cannot regulate your ionic environment, you cannot regulate your plasma osmolality, you cannot regulate your plasma osmolality, you cannot regulate your plasma volume. If you can't regulate your hydrogen ions, you cannot regulate your blood pH. And furthermore, if the kidney starts to fail, you cannot get rid of the metabolic waste. So all of that accumulates, and causes toxicity throughout the whole body. And that's how you die. Okay? So take good care of your kidneys. You got 2 of them, and they're both highly functional. All right, so the kidneys themselves, mainly in your retroperitoneal area, and there's 2 long tubes called the ureters, and the ureters are connected to your kidneys. And the ureters descend all the way down into what we call the pelvic cavity. And as it descends into the pelvic cavity, it will attach to what we call the urinary bladder. The urinary bladder is an organ that's located in the pelvic cavity, and it is the most anterior organ in your pelvic cavity in both male and females. Okay? And of course, from the urinary bladder, which is a temporary storage of urine itself, there is another tube that is connected to that, and we call that the urethra. This urethra allows for you to evacuate or get rid of the urine that is being held inside the urinary bladder for a temporary amount of time. Okay. So these are the 4 structures that basically make up your urinary system. Here's your kidneys. This is on the right. This is on the left. Right kidney is slightly lower in position than your left kidney due to the fact that the liver is on the right upper quadrant. So the liver is a huge singular organ that kind of pushes down, so the kidney has to be a little bit descended or a little slightly lower than the left kidney just to compensate for that massive liver. Okay? And here's these 2 long tubes that connects the kidneys all the way down. These are called the ureters. Okay, and down here, the anterior-most organ within the pelvic cavity, that's the urinary bladder, huge muscular organ, right? It's able to expand to accommodate volume and also contract back to basically, become a little less volume accommodated. And then here's this tube called the urethra, which allows you to get rid of the urine that is being stored inside the urinary bladder. Okay. This is basically some kind of angiogram, or not really angiogram. You put a dye, or you ask the patient to swallow some dye, or you inject the dye into the blood actually, and then from there the blood gets filtered into the kidneys, so then you can see all of this fluid that is draining out of the kidneys into your ureters and eventually into your urinary bladder. Okay. Just kind of keep in mind that the urinary bladder sits inside the pelvic cavity, but as you fill it with urine, you start to fill it from the bottom up, so therefore the urinary bladder starts to expand upward. Okay. So therefore, if you continue to fill with fluid and don't go to the bathroom to evacuate that fluid, then the dome or the top of the bladder basically extend into the lower portion of your abdominal cavity. Okay, abdominal cavity is basically, if you take your draw a line slightly underneath your umbilicus, right, where that waistline should be, then below that is your pelvic cavity, above that is your abdominal cavity. All right. Kidneys themselves, if you take a look at the structure, hopefully you guys remember this from anatomy, if you take a look at it and you slice into it, there's 2 distinct regions. The outer region is known as the renal cortex. The cortex is always indicative of the outer region. Renal refers to kidneys. Okay, so the outer region of the kidneys is referred to as the renal cortex, whereas the inner region of the kidney is referred to as the renal medulla. So this is what it should look like when you cut into this kidney. So the outer portion is out here, right? That's the renal cortex. And then the inner portion is all of this structure in here called the renal medulla. And within the renal medulla are a bunch of these sort of like inverted triangular structure, which is actually a 3-dimensional structure, and we call those the renal pyramids. Okay? And the renal pyramids are basically separated by these tissue here called the renal columns. Right? Now the apex of the pyramids all point toward the center, this indented portion of the kidney here. So here's the apex of one pyramid, here's an apex of another, the apex of another, the apex of another. The apex of these pyramids are referred to as the renal papillae, and they all point toward a collection point, and this collection point is referred to as your minor calyxes. So all the urine that's being created in your kidneys through the filtration process basically will be deposited into these minor calyxes from the apex of your renal pyramids. Okay? From each of these minor calyxes, all that urine will be collected into a larger reservoir called the major calyx. So there's like 4 major calyxes and stuff like that, and then each of those major calyxes, all the urine is deposited into this larger reservoir called the renal pelvis. From there, the renal pelvis kind of converges and becomes this long tube called the ureter. So that's where urine is going to be collected, and it's going to be taken all the way from the kidneys to your urinary bladder. Okay. Just understand the kidney has a huge amount of blood supply because it is basically one of the main structure, one of the main organs that is acting with the blood to help filter the contents of the blood itself, right? And as it filters the blood, the amount of filtration, so it receives roughly about 625 mils per minute. So as far as the overall blood flow, right? And the amount of blood that's dedicated to the kidneys is roughly about 625 mils per minute. Of that amount of blood per minute time, about 125 mils of that blood is filtered through the kidneys per minute time. Okay, so you can imagine that, right? So if the kidneys filter the blood at a rate of about 125 mils per minute throughout a whole 24 hours, then the kidneys will filter roughly about 180 liters of fluid, okay? Or roughly about 180 liters of blood. Now, of that 180 liters, we only lose about 1.5 to 2 liters as urine. The vast majority of it is taken back into the blood. Make sense? Imagine if you lose 180 liters. I think we talked about this once upon a time, right? I think. Maybe it was a long time ago. Perhaps you were listening. But if we lose 180 liters of fluid every single day, we'll be dead by the end of the week. Does that make sense? 180 liter fluids. Can you imagine drinking 180 liters every single day? There's no way. It's impossible. Right? Anyhow, so the kidneys does a very wonderful job to take back most of the contents of that fluid. So we don't lose the water, we don't lose the sodium ions, we don't lose the chloride ions, we don't lose any of that, kind of stuff. All right. So due to that fact, the kidney has a huge amount of blood flow. So there, it comes directly from what we call the abdominal aorta. So from the aorta, you have these 2 branches called the renal arteries, one going to the right, one going to the left. Okay. And then highly branches as it gets into the kidneys. And you guys don't need to know all these branches. I mean, you should, because you already had anatomy, right? But I'm not going to ask you anything about it. But eventually, they're going to branch into microscopic vessels, and these starts with the afferent arterioles, which then leads into this capillary system called the glomerulus, which then leads out into another arteriole called the efferent arterioles. And from the efferent arterioles, it's going to branch into another set of capillaries called the peritubular capillaries, right? And then from there, you're going to get what we call the vasa recta and all that stuff, which I don't know why it's not even mentioned here. I guess I forgot about it. And then it dumps back, everything dumps back into the veins, taken out of the kidneys, and dumped into your inferior vena cava. Okay, so the kidney is one of those main organs that receive a massive amount of blood every single day. Okay. All right. Now, when we are talking about the functional unit of the kidneys, if you look into the microscopic structures of the kidneys, then the functional unit of the kidney is referred to as the nephron. Okay. The nephron, basically, for let's say each kidney, or maybe each kidneys, we have roughly about like 2.5 million of these nephrons. That's each kidney, so roughly about 5 million total, right, for both kidneys. Now, these nephrons, when they work, and they will achieve all of those functions that we listed out before, right? All of those intended 5 functions that we listed out for the urinary system, but they achieve those functions through only 3 processes. And those processes are either filtration, secretion, and reabsorption. Okay. So filtration, you are filtering the blood to make what we call a filtrate. And the filtrate is basically a term we use to indicate the fluid inside the kidneys. Okay. So while the fluid is inside the kidneys, it is not urine yet, as it's moving within these nephron structure. Once it leaves the nephron, then it becomes urine, okay? But while it's circulating inside the kidneys themselves, inside the nephrons of the kidneys, we still call them filtrate. And filtrate is basically a direct filtration from plasma itself, from blood itself. Okay, it's just like CSF is a filtrate of blood and things like that, right? So same thing here. All right. So filtration basically is a process where you're filtering the plasma to create the filtrate. Secretion is, let's say, as the blood moves out of that area, but you still want to take other things from the blood that you did not filter, but if you want to take that and put it into the filtrate, then you can secrete it directly into the filtrate from the blood itself, okay, by a series of transport mechanisms, okay? So that's secretion. Whereas reabsorption is a process where you're taking things from the filtrate, any substance from the filtrate, and water too, and you take that back into the blood, okay? So filtration and secretion, basically taking things from the blood, putting it into the filtrate, whereas reabsorption is taking substances from the filtrate and putting it back into the blood itself, right? Just kind of understand that. Now, for the nephron, it's made up of two parts. One is this large globular, sort of like rounded structure that is composed of multiple things called the renal or glomerular corpuscle. Okay? And the renal or glomerular corpuscle is responsible for the filtration of your blood. This is where the filtration occurs exclusively within the nephron. Okay? Now that renal corpuscle or glomerular corpuscle, that like bulbous rounded area, is actually connected to a bunch of tubes. So therefore we call them the renal tubules. Each of these renal tubules, basically they have different formation and things like that, but along the way, their overall function is to secrete and or reabsorb the contents of the filtrate. Okay? So we're going to talk about all of this and how it's achieved in a second. So again, here's the kidney. You can see we slice through it. We see the cortex on the outside, the renal medulla on the inside. And then we isolate one chunk of that kidney. We see here's the cortex, the renal cortex on the outside. Right? And the renal medulla, in this case, we isolate one of these renal pyramids of the renal medulla. And you can see all of these yellow squiggly lines and stuff like that. We take one of those guys and we magnify them. And we'll see that this is your nephron. Okay, this bulbous portion right here is called the renal corpuscle, and all of these tubes that are connected to it that have different formation and things like that, collectively we call them the renal tubules. Okay. All right. Questions? Here. The rest of it I'm just going to draw for you. Again, just going to put down the functions of the urinary system, achieved mainly by your kidneys. Regulate plasma, ionic composition, or concentration, wherever you guys want, right? Or the amount of ions within the plasma itself. Two, by regulating the ionic composition, you also help to regulate the plasma osmolality. Okay? And if you regulate the plasma osmolality, you also help to regulate plasma volume, which then equals to some degree of blood volume and things like that, right? And the fourth thing is that while they're regulating one of these ions, one of those more important ions is going to be hydrogen ion. So regulate concentration of hydrogen ions, therefore basically blood pH. Okay. So the kidneys will help regulate the hydrogen ion concentration in the blood and in the kidneys, and therefore also help in contribution to regulation of the blood pH as well. And the fifth thing that's going on with the kidneys is to excrete metabolic waste. Okay. It achieves these 5 functions through 3 transport processes. One is called filtration. 2 is called secretion. And 3 is what we call reabsorption. Please keep in mind this is not absorption, because absorption is taking it in for the first time. Reabsorption is taking it back once you have filtered something. Okay. All right. So the structure of the kidneys. Just draw it out for you guys. So let's say the bulbous portion of the kidney is this big portion here. All right, so it kind of looks like this. The overall bulbous portion of this is called the renal corpuscle. It is composed of an outer capsule and an inner layer of what we call capillaries. Okay, so this is the renal capsule out here, and then feeding blood into the renal capsule are these blood vessels, and as the blood vessels come in, they become convoluted. Okay, so there's a blood vessel that feeds blood into this area. There's a blood vessel that takes blood out of this area. Okay, and the blood vessel that is in here, this is a capillary system we call the glomerulus. So the glomerulus together with the renal capsule forms your renal corpuscle. Oh, need a chair. Okay. That is the first portion of your nephron. Okay. Now connected to this, and I'm going to draw all this out first before I start talking about the functions individually of this thing. The capsule itself become these type of part of what we call the renal tubules, and the first portion of the renal tubules is a convoluted portion of the renal tubules. Right? It's kind of twisted. It's not straight. So just think of it like coiled together and stuff like that. So we call this the proximal convoluted tubules. That's a D. Okay. And then from there, the proximal convoluted tubule starts to straighten out, and it starts to dive deep down this way. And as it dives deep down this way, it has a thick and a thin portion, and then it starts to go back up. And as it goes back up, it becomes thick again. Okay. So this whole thing we call the nephron loop. Otherwise known as the loop of Henle, named after Dr. Henle. Okay. Now, the loop of Henle has a descending portion, which is moving downward. So we call this the descending loop. And an ascending portion, which goes upward. So we call this the ascending loop. Okay. And then from there, the ascending loop becomes another tubular system, which is also convoluted. And we call this the distal. Convoluted tubules. Okay? And the distal convoluted tubules will basically all dump into this long tube that is straight, and it collects from multiple distal convoluted tubules. Okay, so we call this the collecting duct. Sense? Any questions about the nephron? So this is the structure of the nephron, right? It's made up of two parts. The renal corpuscle, which includes your capsule, and the glomerulus, which is a capillary system. And then you have your renal tubules, which starts with the proximal convoluted tubules. Then it basically straightens down into the descending loop and then the ascending loop, together forming the nephron loop or the loop of Henle. Once the ascending loop ascends back up, it becomes the distal convoluted tubules because it's distally located. And then all of these distal convoluted tubules dump into the collecting duct, which collect and basically is oriented all the way to the apex of that pyramid, so that whatever content is in here eventually gets excreted as urine. Okay? All right. Now, another thing that you guys need to know is that I forgot to label these vessels here for you. So blood is gonna enter via this vessel we call the afferent. Let me see where I'm gonna put that. Afferent arteriole. Okay, so blood is gonna enter. Let me just extend that out a little bit. Blood enters into the glomerulus from the afferent arterioles, and blood is going to leave the glomerulus via another vessel we call the efferent arterioles. Okay. And the efferent arterioles will basically form these capillary system. The capillary system here is going to run around or right next to the proximal convoluted tubules, and also right next to your distal convoluted tubule as well. Okay. So all of these blood vessels here, these capillary system, we call them the peritubular capillaries because they're right next to the tubules themselves. The capillaries over there too. Just don't have enough room to write that. So you guys just imagine that guy, same thing here. Good? All right. So in the glomerulus, as blood is being diverted through the glomerulus, right? Blood is basically circulating all the way through, right? This is going to be plasma. So the plasma is going to be filtered, gets pushed out. Okay. How do we filter stuff? You guys remember the Starling principle that we talked about once upon a time? Right? You guys were testing it last week. So it should still be somewhat fresh in your mind. I don't know, maybe the 3-day weekend or whatever has, or 5-day since we last seen each other, has taken a lot of that information out of you. But do you guys remember what are the 4 Starling forces that exist in every single capillary system?
Hydrostatic and oncotic.
Hydrostatic oncotic pressure, right? So therefore, within the plasma itself, there is a hydrostatic and oncotic pressure. But also in this space here, in the renal capsule, there's also a hydrostatic and oncotic pressure as well. So those 4 forces interplay with each other, but it always results in a constant filtration and no reabsorption. Okay, so you guys remember the net filtration pressure versus the net reabsorption pressure that occurs, right? As the fluid moves into the capillary system and all 4 of those Starling forces interact with each other to produce a net effect. Well, in the kidneys, when those 4 Starling forces interact with each other, it will always produce filtration right at the glomerulus. There is no reabsorption going on at the glomerulus. Okay, only filtration, because the overall pressure, the what we call the glomerular hydrostatic pressure overall is going to be higher than all the other pressures combined. So therefore, there's a constant pressure that pushes fluid out of the glomerulus into the space within the renal capsule. Okay, so therefore this whole process here is referred to as filtration. So ask yourself, what are we filtering from the blood itself? All right, so let's say here's your inside your glomerulus, right? So you have blood or plasma, we'll call it plasma. So glomerular corpuscle. Remember the glomerular corpuscle is the same thing as the renal corpuscle, right? So it's just another term for that. Okay, so on this side, you have plasma. Okay, and then the cells that surround this capillaries is basically a fenestrated type of cell. So you have tiny little gaps, little gaps, things like that. Fenestrated capillaries. You guys remember fenestrated capillaries, right? Maybe. I hope. Yeah. Draw tiny gaps and stuff like that on there. Okay. So let's say these are the cells that make up the walls of your glomerulus. So a bunch of fenestrated or fenestration here. Okay. We're not going to talk about what else is in there because there's like 3 things that makes up this particular filtration membrane. But inside the plasma, there's going to be pressure that pushes the fluid out. Now, the content in the plasma, you have to understand, is mainly water, right? It has every single thing that you can think about, gases, any kind of ions, organic compounds like glucose, amino acids, and anything else, right? And also has a lot of big, large proteins, albumin, fibrinogen, globulins, and stuff like that, right? Or globin proteins, sorry, that basically transport those fat-soluble vitamins. So now, if you have a series of filtration, so you're filtering the blood, what are you creating on this side? So then the fluid component in the space here in the renal corpuscle is referred to as filtrate. So what does filtrate contain? Water. What else? Ions. Whatever ions you want. Take your favorite 5. Right, sodium ion, potassium ion, chloride ion, bicarb ion, calcium ions, hydrogen ion, right? Doesn't really matter. What else? Organic compound. Like glucose and amino acids, okay? Does it have any blood proteins? We wouldn't call them blood proteins if it stays in the kidneys, right? It's called blood protein because it stays in the blood. And the reason for that is because this area here, with all these structures that form that membrane, it is restricting the movement of the blood proteins. So all your blood protein stays in your blood. So therefore, let's say if you're doing some kind of, I don't know, sort of like urinary analysis, right? Urine analysis and stuff like that with the patient, and you see that there's proteins inside the patient's urine. Well then, something's going on with the patient's kidneys, especially right at this particular area called the renal corpuscle. And of course, the thing that really is affected the most will be the cells that forms the glomerulus itself. So something has to be happening, otherwise you wouldn't be leaking large proteins, because the large proteins will not be able to fit through those fenestrations, okay? What if you do a urine analysis and you see a bunch of red blood cells in the urine? Something's going on there as well. Okay, some kind of destruction, some kind of inflammation, maybe some kind of autoimmune diseases and stuff like that. Lupus, diabetes, whatever have you, will affect that area of the kidneys. Okay. Now, what if you check and you see a bunch of white blood cells? Again, something is going on in that area of the kidneys because the cells are much larger than the proteins. So if the cells can squeeze through, that means some destruction has already been happening inside your kidneys. Okay? So it could be some kind of ascending urinary tract infection that starts in the urinary bladder and ascend all the way up to the kidneys and now causes some destruction or some kind of toxicity inside the kidneys that basically destroy a bunch of these particular nephrons, and therefore now you're just leaking a bunch of blood into your filtrate. Okay? So therefore, the filtrate should only contain these compounds. And the main ions we're really concerned about is going to be sodium ions, potassium ions, chloride ions, calcium ions, and things like that, right? Hydrogen, we'll think about hydrogen as far as pH imbalance later on. Organic compounds, we're mainly concerned about glucose and amino acids. Okay, so that's the point of filtration. So now you have all of this entering the proximal convolute tubule. So filtrate will enter here. Once the filtrate enters the proximal convolute tubules, now you have to do something to either take these contents back into the blood or lose them forever. Okay. Now you can also take some contents in the blood if you have an excess amount of those, and you can put it into the filtrate. So therefore, if you have not filtered it in the glomerulus, and whatever those contents are now in your peritubular capillaries, you want to take any of those, let's say ions or whatever it is, and you want to take that and put it into the filtrate from this area, put it into the proximal convolute tubules, then we'll call that process secretion instead of filtration, because you're no longer filtering anything, you're just transporting these across the membrane. So therefore we call this secretion. Okay. Any substance from the filtrate that you take back into the peritubular capillaries, we will refer to as reabsorption. Okay. So at the proximal convolute tubules, there's a series of activities called reabsorption and secretion that takes place. And the reabsorption and secretion that takes place at the proximal convolute tubules and these peritubular capillaries are unregulated, which means you don't need any external forces to help that process. There's no hormones involved in the regulation of the reabsorption and secretion that goes on, right? So therefore, it's called unregulated reabsorption and secretion. All right, so let's say in the proximal convolute tubules, okay, imagine you have these cells here, and then here's your basement membrane. Imagine here's your blood vessels down here. Okay, here's your peritubular capillaries. Okay, these are the cells of the proximal convolute tubules. Here's the filtrate that enter. Okay, within this filtrate, you have a lot of water. We're concerned with sodium ions, we're concerned with chloride ions, we're concerned with potassium ions and things like that. We're also concerned with glucose and amino acids, right? Because those things we don't want to lose. If we lose amino acids, you just wasted all of that. Now you have to consume it or make new ones, right? Because amino acids are a very essential building block of your protein structure. And glucose is your primary carbon sources that every single cell used to make ATP. So you don't want to lose any of that stuff. All right, so now how do you get these contents to be taken back into the blood? Well, there's a driving force for all of this, and it's located on the bottom of these cells. Okay, and this particular structure here is a primary active transporter called the sodium-potassium ATPase. And the sodium-potassium ATPase is going to take 3 sodium ions and pump this out into the interstitial space so that it can move into the peritubular capillaries. It's going to take 2 potassium and it's going to pump it into this cell. Okay, and those 2 potassium, the potassium basically came from the blood that has basically moved out into the space. Right? So therefore, you're actually taking potassium from the blood and putting it into this cell, and you're going to take the sodium ions in this cell and pump it out back into the blood. Now, when you're constantly doing this, you are using ATP, because this is what we call a primary active transporter. Okay? Using direct energy of ATP, it's going to move these ions against their respective concentration gradient. It's moving sodium ions from an area of low concentration inside the cell to an area of high concentration outside the cell. It's going to move potassium ions from an area of low concentration outside of the cell and move it into a higher concentration of potassium ions inside of any cell. Okay? This creates a concentration gradient of sodium ions and potassium ions. Okay? So if that's the case, on the apical surface, on this surface of the cells that makes up your proximal convoluted tubules, are a bunch of sodium ions. So immediately, as you create this concentration gradient and this filtrate enters the proximal convoluted tubules, the amount of sodium ions in here will be higher than the amount of sodium ions inside your cell. So therefore, you have an immediate facilitated diffusion of sodium ions back into this cell. And when this happens, you can see that this particular sodium potassium ATPase constantly pump the sodium ions out, and therefore sodium is now progressively being reabsorbed back into the blood. Make sense? Okay. Some of these potassium may also go back through other mechanisms of transport. Now there's a trigger that basically allows for chloride to also be reabsorbed back into this, and then there's a chloride channel here. So we're not gonna demonstrate too much how chloride gets reabsorbed. It's just basically a bunch of facilitated type of diffusion activities. Okay. So anytime sodium ion gets reabsorbed, that's a trigger for chloride ions to also be reabsorbed. Now, anytime sodium and chloride gets taken back, water also follows. Okay. Now, water is going to be taken through the cells, but some of them will squeeze through between the cells. I'm just drawing this for simplicity's sake. Okay. So therefore, as sodium ions and chloride ions are taken back into the blood, so you've reabsorbed sodium ions and chloride ions, you've also now reabsorbed water as well. Okay. You can reabsorb potassium ions as well, or you can secrete it out, and you can also secrete out hydrogen ion, because there's an exchanger here. Basically, it's an exchanger that as sodium ions gets taken in, hydrogen ion is taken out. Okay. Then some of these particular ion channels here allow for this potassium ions to basically go back into the blood as well. Okay. So essentially, what have we reabsorbed at the proximal convoluted tubules? We've reabsorbed water, which is one of the most important things, remember, to regulate the blood plasma volume. But we've also reabsorbed sodium ions. You can clearly see it's moving back into the blood. We've reabsorbed chloride ions, moving back into the blood. We've reabsorbed water. We've also reabsorbed potassium ions, but we have secreted, essentially, hydrogen ions. Okay, so now we've taken hydrogen ion, let's say, from this cell, and then we have just taken it out and placed it into the filtrate. But of course, in addition to that, we have also filtered some amount of hydrogen ion as well. Okay? So that's the reabsorption and secretion process that's going on. Keep in mind, it is unregulated by hormones or anything else. Okay? This is just a self-regulated mechanism due to the fact that the sodium potassium ATPase, which is located on the basal side of the cell, is constantly working. If you consume something in, let's say, medications or food or whatever the case is, that poisons this primary active transporter, then the kidneys will shut down. Okay? All right. Now, how about the glucose and amino acids? How do we take those back into the blood? So let's say that's another cell right next to this one. And on the surface of this cell, you have this particular series of exchanger. Again, this is driven by your sodium potassium ATPase, right? So taking sodium ions, pumping it here, and then moving it into the blood. Taking potassium ions out of the blood, and then pumping it into these cells, okay? Just to create a concentration gradient. So that's okay, so that's ATP. All right. Now, on the apical surface of these cells, once again, there's this exchanger protein that allows for sodium to be taken in, okay? But this also allows for glucose to piggyback that and being taken into this cell. Then glucose itself will be transported progressively back into the blood. Okay. Whereas there's also for amino acids, there's also another exchanger that allows for sodium ion to diffuse into the cell. As the sodium ion diffuse, it's going to allow for amino acid to piggyback that as it's taken back into these cells. And the amino acids will have their own dedicated transport mechanism at the basal portion, which then take them out into the interstitial space and take them back into the blood. Okay? And therefore, at the proximal convoluted tubules, we have also essentially reabsorbed glucose and amino acids. Okay? Now, how much of the water, the sodium ions and the chloride ions, how much of those contents has been reabsorbed at the proximal convoluted tubules? Of all of the contents of the filtrate that has been filtered at the glomerular corpuscle, right? So imagine this whole filtrate is like 100%. That 100%, as it moves through the proximal convoluted tubules, 85% of the water, the sodium ions, the chloride ions, and somewhat the potassium ions has been reabsorbed back into the blood. Okay? So 85% of these contents have been taken back into the blood or has been reabsorbed back into the blood. With the glucose and amino acids, 100% of those has been reabsorbed back into the blood. Okay? So for these guys, so at the proximal convoluted tubules, reabsorption and secretion going on. Okay, so reabsorption, we'll say 85% of water, sodium ions, chloride ions, and so on. Let's just focus on those guys. Okay, and then for the reabsorption, it's 100% of your glucose and amino acids. Secretion, we secreted mainly hydrogen ion. That's the main thing that we secreted from the proximal convoluted tubules. Make sense? Okay. So of course, this is like a very gross summary of what's going on inside the proximal convoluted tubules. Okay. So now let's say this. I know we didn't have a lecture talking about membrane transport because we didn't have time, but for the transport of glucose and amino acids, those falls into the category of secondary active transport. In secondary active transport, because the cell already has glucose and amino acid in somewhat abundancy or higher concentration than the glucose or amino acids in your filtrate. But you still want to take back all of that glucose and amino acid because you don't want to lose any of these in the urine. So therefore, you have to transport these in an uphill manner, like actively transport the glucose into the cell and actively transport the amino acids into the cell. In order for the cell to do that, it uses the diffusion activities of sodium ions that is set up by your sodium potassium ATPase. Okay? Because by this primary active transport working, you are now lowering the amount of sodium ions inside the cell. And by lowering the amount of sodium ions inside the cell, you allow for sodium to diffuse passively back into the cell. But these particular proteins here, they can use that diffusion activities. And they use what we call the diffusion energy. As sodium diffuses back in, they take that energy and they allow for the glucose to be reabsorbed or to be taken back into the cell in what we call an active manner, going from low glucose concentration out here to a higher glucose concentration inside the cell. Okay? And so on and so forth. All right, so now let me ask you, clinically speaking, what if you decide to have, I don't know, wake up one morning, you're like, I just don't feel like
drinking water, I just wanna drink maple syrup. I don't know, just an experiment, right? Let's say you drank like a whole gallon of maple syrup throughout the day, or like just in the morning, then you go to school and you're like, oh, okay, I'm, super, excited now because of that sugar rush or whatever the case is. Then you're gonna start to overwhelm those secondary active transport, right? Because they have some degree of like active sites that they have to bind to, so they'll reach some form of saturation at some point and stuff like that. Once you've saturated all these carrier proteins, what's gonna happen to the excess glucose and amino acid? It's gonna continue, right, in the nephron, because the filtrate continues past that, all the way through these particular tubules. Now, along the way, there's no other site where glucose and amino acids gets taken back into the blood, okay? The glucose and amino acids are mainly taken back, if not all of them, are actually taken back at the proximal convoluted tubules. So you cannot take it back from the proximal convoluted tubules. All the excess glucose and amino acids will end up in your urine, okay? Now, how can you tell if you have an excess of glucose in your urine? Color doesn't really tell you much. Because you could be drinking a lot of water too, right? What? Say it.
Taste.
Yeah, the taste is sweet. So how can you test whether or not you have glucose, excess amount of glucose in your urine? If you don't have a urine dipstick, right? Obviously, you can buy a kit from like Amazon or whatever. Like they can expedite the shipping. It would be here before you guys get home and stuff like that, right? But what if we don't have that luxury?
Be outside and ants will like pretty much eat your urine.
Oh, you know. So you can do that. Go home and do that tonight. Everybody go pee in a cup. If you want to know if you have excess amount of glucose in your urine, aside from tasting it, I mean, it's your own urine, right? Who's gonna judge you? I mean, we're not telling you like gulp it. We just like dip your finger and then just, all right, it's tastes like urine. I don't know what urine tastes like, but if it's sweet, then there's a little bit of that kind of like sweet aftertaste, right? But if you don't want to do that, just go pee in a cup, leave it outside. Well, maybe not tonight because it might rain, right? It might dilute your urine. Do some other night when it's not raining. Just leave it outside and then check it first thing in the morning. If you have a bunch of ants congregating, singing kumbaya around your urine, then you know you have some excess amount of glucose in your urine. Okay, it's just a quick way to check. Of course, you can go to your nearest, pharmacy or clinician or whatever and say, yeah, can you do a test? They can test your urine, they can test your blood and stuff like that to see if you do have that. Now, if that happens one time, it's not a big deal, okay? But it happened chronically, then it puts you in a state of what we call diabetes. Okay, so there's obviously type 1 versus type 2 diabetes, but that's all more like hormonal and genetic components associated with that. But anyhow, if you do consume an excess amount of sugar all the time, you're gonna overwhelm this transport mechanism and you will have some amount of sugar all the time in your urine. And that's not always good. If you have an excess amount of glucose in your urine, that means you already have an excess amount of glucose inside your plasma. So, all right, all of this happening in the proximal convoluted tubules. So we're gonna have reabsorption. Okay, of substances from the filtrate back into the peritubular capillaries. And we will also have secretion. Of substances from the peritubular capillaries into the filtrate, right? Through those processes that we just demonstrated. Okay, now once the filtrate enters or leaves the proximal convoluted tubules and enters what we call the descending nephron loop, there's only 15% left of the total amount of filtrate that has been filtered by your glomerulus. Because remember, 85% has already been taken back into the blood at the proximal convoluted tubules. So with that 15%, then the rest of these renal tubules of the kidneys is gonna fine tune the contents of that filtrate. If you don't need to take anything back, then it's not gonna take anything back. If you do need to take something back, then it will do something to take that back, okay? So imagine now your filtrate enters this descending nephron loop. Now within the filtrate, you're gonna have water, and you're gonna have, let's just put the main ions, sodium ions and chloride ions. You shouldn't have any more glucose or amino acids by the time you get here, okay? However, once you get into this descending nephron loop, the cells of the descending nephron loop will only allow for water to be reabsorbed, but prevents the solutes, these sodium ions and chloride ions, from being reabsorbed, okay? So water is permeable at the descending nephron loop, but the solutes are not permeable at the descending nephron loop. Okay, so solutes cannot get through. It's only the water that gets reabsorbed. Okay. All right, so if the water is being taken back, and this filtrate continues all the way to the bottom of this loop, what's going to happen to the osmolality of that filtrate? Is it going to be super high or super low? Yeah, it's going to be high because the water has essentially been taken out, and we're not talking about all the water, we're talking about like the vast majority of the water. So therefore, what's left is a bunch of filtrate, right? Or it's a bunch of solute. So therefore, if you have more solute than the amount of water, then it becomes highly concentrated, and therefore we have a very high osmolality. Okay. All right. Now that filtrate continues at the bottom of the loop, and it's going to be taken back up into the ascending nephron loop. So in the ascending nephron loop, again, the filtrate enters here. And the filtrate again has water, sodium ions, chloride ions, right? Now in the in the ascending nephron loop, the water is impermeable to these cells, or these cells prevent the water from being taken back. But these cells allow for the reabsorption of sodium ions and chloride ions, right? So again, from the descending nephron loop, you have water being taken out, and essentially will be taken back into the blood. From the ascending nephron loop, you have the solute being reabsorbed, and eventually will go back into the blood. But while the solutes are being taken out into the space here before it gets into the blood, it actually now raises the osmolality of this space. So the space between the descending and ascending nephron loop becomes what we call hyperosmolar or hypotonic, right? Compared to the contents inside the descending nephron loop. Because now you have a bunch of filtrate, or you have a bunch of solutes being taken out here before it gets back into the blood. So then the amount of solutes that is being reabsorbed at the ascending nephron loop is going to influence the amount of water that gets reabsorbed at the descending nephron loop. Does that make sense? So imagine you have the first set of filtrate coming in, right? Some amount of water is taken out. So now it becomes very highly concentrated in your solutes. And the solutes continue to go up this way, and the solutes gets taken back. So therefore, the next set of filtrate as it come in, now the space out here has a lot more solute. So it becomes a little bit more hyperosmolar or hypotonic compared to the filtrate that's coming in. So because of that, water is gonna get taken out. Water is gonna get pulled out. Why is that? Why is water basically being influenced to be reabsorbed more so if you have a hypertonic environment? Because how does water move?
Area of high concentration to lower.
Going from high concentration lower of what?
High concentration of water to low concentration.
We don't care about water concentration, we care about solid
Low solids
concentration.
to high solids.
So now water is gonna be more influenced to move from area of low solid concentration in here to a much higher solid concentration out here, right? Because remember, the first set of filtrate that goes through, the water is already taken out, so therefore you have a bunch of solutes, so it becomes super osmolar, hyperosmolar here, and as it comes over here, that high degree of that solutes will be taken out into the interstitial space. That immediately raises the osmolality of the interstitial space, so therefore creating a very hypertonic environment in here, which means the next set of filtrate as it continues in, now you have much higher concentration of solutes here in the interstitial space compared to the amount of solutes that's inside that filtrate. So immediately water is gonna get pulled out drastically. Okay, so this happens continuously. And of course, it's also aided by blood flow and stuff like that, which we're not gonna, overlap in this particular activities. But imagine so you have two loops, the descending loop, which is the first portion, ascending loop, which is the second portion. The activities of the second portion influence the activities of the first portion. So therefore, the reabsorption of the solutes from the ascending nephron loop is going to enhance the reabsorption of water from the descending nephron loop. Because water is essential. It's very important, and we need to conserve that water as much as possible so we don't lose it as urine. Okay? So this process here is referred to as the counter current multiplier system. And this counter current multiplier system also depends on the flow of blood as well. Okay? But essentially, it's just a very basic way to think about it is that the latter portion, the second portion of this loop is going to influence the activities of the first portion, which means the ascending loop, the activities of the ascending loop to allow for the solutes to be reabsorbed is going to increase the reabsorption of water from the descending nephron loop. Okay? And this is constantly working, but it kicks in furthermore, it kicks in a lot better or in higher gear when you are volume depleted in your body. Okay? So if you're volume depleted, that means you're dehydrated. So if you're dehydrated, then the amount of blood that gets into the kidney is decreased. And if that's decreased, the amount of filtration is decreased. That means the overall amount of filtrate is already low. 85% of that low filtrate will already be taken back from the proximal convoluted tubules back into the blood. So whatever is left is already very, very low. So therefore, it is detected by that, and so therefore, these particular countercurrent multiplier systems start to work harder. Why? Because you now want to take even more water back into the blood, because you don't want to lose that. Okay. So countercurrent multiplier system is a great system to basically ensure that you don't lose an excessive amount of water from the nephron itself. Okay. To ensure that the water that is being delivered by this filtrate into this descending nephron loop is constantly taken back into the blood all the time. Just maximally conserve that water as much as possible. And of course, you're also taking back the sodium ions and chloride ions, so you don't lose that as well. Okay? All right. Any questions about the countercurrent multiplier system? Okay. Now, as the filtrate continues, so let's say after all this is done, whatever filtrate is left, right, enters the distal convoluted tubules. Okay? As the filtrate enters the distal convoluted tubules, again, there's a series of reabsorption and secretion that occurs. So we still have reabsorption. And we also have secretion going on here. Okay? So whatever content is left, you can actually now take back some of those contents back into the blood. And if there's any waste material inside the blood that you don't want, that you haven't secreted over there, you can now secrete it in the filtrate here. But the process of reabsorption and secretion that occurs at the distal convoluted tubules are highly regulated. And it will be regulated by hormones. Okay? So at this particular point, the kidneys will decide if there is an inadequate amount of substances in the blood, or you have to basically take it back into the blood, then through hormonal regulation, the kidneys will then reabsorb and secrete these substances. Okay? So how does it do that? So let's go over here and take a look. All right, so distal convoluted tubules. Right? So here's a cell. Okay, and again, at the bottom of the cell, there's the sodium, potassium, ATPase. Okay, but at this point in these cells, the sodium, potassium, ATPase is not as highly active as the sodium potassium ATPase in the proximal convoluted tubule. So here's still your peritubular capillaries. Okay. Your filtrate is flowing through. Okay, so in your filtrate you have water, you have sodium ions, you have chloride ions, and things like that. But there's nothing on the apical surface of these cells, or at least not as extensive as those guys, to take these substances back unless the kidneys tell them to take it back. And the only way the kidneys can tell them to take this back is to release adequate amount of hormones. So therefore, on the distal convoluted tubules, there are a series of cells that are located here. And these are called the macula densa. So you have a bunch of cells called the macula densa, and once the macula densa detect this filtrate as it courses all the way into the distal convoluted tubules, the macula densa will respond to those filtrate. Okay. Now, what the macula densa detects is the volume of the filtrate and the concentration of the solids within that filtrate. If the volume is low, or the amount of concentration of these solutes are also low, then the macula densa will start to respond aggressively against that. Okay? There's another set of cells lining the afferent arterioles for the most part. It also lines the efferent arterioles. But let's say these cells line the afferent arterioles more. And we call these the granular cells. So hopefully this starts to look familiar to you guys at some point, right? Because we talked about this when we talked about the cardiovascular system. Okay? So when you have in the kidneys, so let's say around that particular area, right, distal convoluted tubules, they have these macula densa. And what the macula densa detect is going to be low filtrate volume and low solute concentration inside the filtrate. Or basically, you think about it, it just detects filtrate volume and detects solute concentration. But if that filtrate volume is lower than what it likes it to be, or the amount of solutes inside that filtrate is lower than what it should be, then it will trigger the macula densa. So therefore, the stimulus to the macula densa would be low filtrate volume. Oops. So that's the first trigger. The second one is low solute concentration within the filtrate. Okay. So when the macula densa detects this, it will release some kind of signal that will stimulate the granular cells. And the granular cells will secrete a hormone we call renin. Bells better be ringing in your head. If they're not ringing in your head, I don't know. Turn it on. What is renin?
To the liver.
To the liver. Good. And at the liver, what does it do?
Triggers angiotensin.
Angiotensinogens are right there.
Oh, and
it doesn't trigger
angiotensin.
anything, but angiotensinogens
Yeah.
there, right? So the liver made a bunch of this. So what does the liver, what does renin do to angiotensinogen? So it converts it into angiotensin 1, not 2.
Turns 1 into 2.
What turns 1 into 2? Renin doesn't do that. Now, angiotensin 1 is going to be secreted by the liver. And as it's secreted by the liver into the circulation, eventually angiotensin 1 is converted into angiotensin 2. By what?
Angiotensin converting enzyme.
by an enzyme called angiotensin converting enzyme, which is mainly located in the lung tissue, of all places. Okay? Angiotensin 2 is a very powerful hormone. Now, what does angiotensin 2 do?
Stimulate antidiuretic hormone.
Okay, one of the things that it does. So, here, you say, 1, stimulate what to release antidiuretic hormone? Stimulate secretion of antidiuretic hormone from where?
Supraoptic nucleus.
From the posterior pituitary gland. Where is antidiuretic hormone made in?
Supraoptic nucleus.
Supraoptic nucleus of the hypothalamus. Okay, so it's made in the hypothalamus, but it gets transported and stored inside the posterior pituitary. So therefore, when angiotensin 2 is secreted, it can stimulate the posterior pituitary to just release that antidiuretic hormone. Okay, good. What else does angiotensin 2 do?
Release aldosterone.
Release what?
Aldosterone.
Aldosterone from where? And we didn't say which part of the adrenal glands, right? So you guys don't have to worry about that. We will in the following something. We'll talk about it in the endocrine system. So stimulate secretion of aldosterone. From your adrenal glands, right? Good. Okay. So, in this particular case, aldosterone gets taken into the blood and is delivered all the way to the kidneys. Okay. It gets taken all the way to the distal convoluted tubules, where it basically, because it's a type of hormone we call a steroid hormone, and steroid hormones are non-polar for the most part. Aldosterone can just move straight into this cell. Once inside the cell, aldosterone is going to stimulate the sodium potassium ATPase to vastly activate this guy. So once the sodium potassium ATPase is activated, what's it gonna do? It's gonna take 3 sodium ions, and it's gonna pump it out. It's gonna take 2 potassium ions, so 3 sodium and 2 potassium ions, and pump it in. So essentially activating the sodium potassium ATPase at the base of this cell. Okay? Furthermore, aldosterone will stimulate, get all the way into the nucleus of the cell, and stimulate the production of ion channels. So it's gonna place a bunch of ion channels on the cell, and one of those ion channels goes here, and what it does is that it's gonna take sodium ions, it's gonna pump it in, or it's gonna diffuse it in, allow for sodium ions to move in. It's also going to allow for 2-chloride to also move into the cell, and this also allow for potassium to also be pumped back in as well. Okay? And at the same time, some of this potassium ions have their own ion channels, which basically allow them to go back up into this filtrate. It's kind of weird. So therefore, anytime you start to move sodium and chloride, again, water is just gonna passively follow. Okay. So what is the purpose, what is the function of aldosterone at the distal convoluted tubule? So here, let's say aldosterone goes to the distal convoluted tubules. So that's the target, right? And what does it do at the distal convoluted tubules?
Yeah.
So 1, increase reabsorption of sodium ions and chloride ions. Okay, so it activates this ion channel to basically allow for the cell to take the sodium and chloride back into the blood. Okay. And at the same time, as sodium and chloride moves back into the blood, water will passively follow. So, passively reabsorb water. But keep in mind, aldosterone does not do this directly. It's just a side effect of the sodium and chloride movement back into the blood. Okay? The second thing that aldosterone does is that it will increase secretion of potassium ions. So you can see that it allows for potassium ions to basically leave this particular cell because it also stimulates the expression of these potassium ion channels here. So that's the function of aldosterone at the distal complement tube. You guys remember you have to make that list, sort of the one I asked you to make, right? That kind of outline all of this. So what do we call this whole system?
Right.
Call it the renin,
Angiotensin,
angiotensin,
aldosterone.
right? Aldosterone system. Or the RAS. Okay. What is the structure here that basically collectively is made up of the macula densa and the granular cells? What do we call that?
Juxtaglomerular apparatus.
Okay. It's an apparatus because it's made up of multiple things. Juxta means next to, glomerular refers to the glomerulus, right? Or this whole glomerular corpuscle. So next to the glomerular corpuscle, and it's made up of a collective different amount of cells. So therefore, the macula densa, the granular cells, and other things makes up your juxtaglomerular apparatus. The macula densa will detect the filtrate volume and filtrate concentration of solutes, and if those two are low, then it triggers the overall renin angiotensin aldosterone system. And of course, when it does that, the reabsorption now is highly tightly regulated by hormones, and that hormone mainly is going to be aldosterone. Okay. There's another hormone that is released out called parathyroid hormone that also acts at the distal convoluted tubules to reabsorb calcium ions back into the blood when you have low blood calcium ions. But we'll talk about that later when we get to the endocrine system. Okay. Any questions about the regulated reabsorption of, or regulated reabsorption and regulated secretion of substances at the distal convoluted tubules? Let me underline the distal convoluted tubules. Okay. Oh, I might as well just put this here. So this is called unregulated reabsorption and secretion. This is called regulated. And it's regulated by hormones. Okay. So regulated reabsorption and secretion. What is it reabsorbing? It's reabsorbing sodium ions, chloride ions, and water. And I'm gonna put a little asterisk on the water because it doesn't, the water gets reabsorbed, but not under aldosterone influence. Okay? And what does it secrete? It secretes potassium ions. Keep in mind that in addition to aldosterone doing this, angiotensin II can also do this at the distal convoluted tubules. Okay, so both of these hormones working together at the distal convoluted tubules, basically trying to reabsorb as much sodium, chloride, and water as possible. Okay. But it's only aldosterone that can actually secrete potassium ions. Angiotensin II can't do that. All right, good. Questions? Last place. There you go.
So now, once you go through this regulated reabsorption and secretion of all these substances, right, whatever's left gets taken into the collecting duct. So now as it moves into the collecting duct, this is filtrate that has things like water and a bunch of solids, right? So we just put solids and sodium, chloride, and other stuff. And you have waste material in here, so right? Waste material being transported all over the place. Now, as a last ditch effort of your kidneys to take back and conserve as much of this water as possible, at the collecting duct, the cells that line the collecting duct can also be influenced by hormones. Okay, so let me just kind of draw a little bit of it here. We don't have any more space over there. So imagine these are the cells in your collecting duct. So collecting duct. Okay. Now in the blood, there's a hormone called antidiuretic hormone that gets released out by your posterior pituitary, right? Antidiuretic hormone is gonna go all the way inside, or gets all the way to the collecting duct, and it's going to stimulate the collecting duct. It can't get into the cell because this is not a steroid hormone, but it stimulates the cells of the collecting duct. And what happens is that the collecting duct will start to place water channels onto the surface of these cells. What do we call the water channels? Aquaporins. Good. Okay, so it starts to place a bunch of aquaporins on the surfaces of these cells, so therefore it allows for massive amount of water reabsorption. Okay, so antidiuretic hormone is going to stimulate what we call water reabsorption at the collecting duct. Why are we doing all that? Why is the renin-angiotensin-aldosterone system doing that? Because of what?
Maintain blood volume.
Why? What's the stimulus that triggered them to do that?
Low map.
Low filtrate volume and low solute concentration, right? Low map, they don't care about that. They don't know if there is a low map or not, right? But if there is a low map, let's say pressure overall inside the blood. If you have low pressure inside the blood, what's gonna happen to the filtration activities? Higher filtration or lower filtration?
Lower.
Yeah, because filtration is mainly driven by your hydrostatic pressure, right? And the hydrostatic pressure is sort of like an extension of your mean arterial pressure, right? The mean arterial pressure pushes fluid in, so flow equals delta P over R. Remember Ohm's law, right? So therefore, if you have adequate amount of pressure, you will have adequate amount of flow. If you have adequate amount of flow, you're gonna have a high filtration, and therefore you have more filtrate to do what you need to do throughout all these areas. But if you already have low amount of blood volume because, let's say you're dehydrated, you don't have any volume, or your volume depleted because you're losing volume somewhere, then the filtration that occurs at the glomerular corpuscle decreases. That means the amount of filtrate also decreases. Therefore, you've taken back 85% here, so therefore this filtrate is actually even less now than before, right? The countercurrent multiplier system kicks in even more because it sensed that, oh man, we have low water and low filtrate, so I'm gonna take back the filtrate, I'm gonna take back the solids so that we can take back more water. So the countercurrent multiplier system is actually already amped up at this point. So by the time whatever is left of that filtrate reaches the distal convoluted tubules, it is actually much lower in overall content and volume than what the macula densa would like it to be. It should already be low, but now it's actually even lower than that. So the macula densa says, wait a minute, why is it that the filtrate that is coming to me is so low? I don't like that. So what I'm gonna do is to increase that filtrate. How do I increase the filtrate? Well, it has to start with the plasma, because the plasma through filtration gives you the amount of filtrate. So therefore, the macula densa and all the juxtaglomerular apparatus structures will think, well, in order for me to get more filtrate back to me, I need to raise the plasma volume, because that is the thing that drives the filtration process that makes more filtrate. So how do I do that? Well, I have this wonderful hormone called renin. I'm gonna release that out. It's gonna convert angiotensinogen into angiotensin 1, which then get converted into angiotensin 2 by this enzyme in the periphery. Angiotensin 2 then kicks off all the other release of other hormones. And therefore now you have this powerful hormone and this powerful hormone to act at the distal convolute tubules to stimulate those cells to take back more solids and more volume. Okay, so at the distal convolute tubules, you're actually now taking back sodium ions and chloride ions and water. Okay, so now the filtrate, which is already low, as it leaves this area, it's going to be even much lower than before. But it doesn't matter, because you're taking this substance back into the blood, because you are volume depleted. And we don't care about making urine at this point. We only care about conserving the contents in the blood at this point. Because who cares if you can make urine when you don't have enough blood to basically provide oxygen to your cells, right? Hey, great, you can pee, but now you're gonna die, okay? So therefore, the kidney's like, screw that. We're gonna serve as much of this volume back into the blood as possible. We don't care about making urine at this point. So now this process is driven massively by these hormones, so that you have regulated reabsorption and secretion of these substances. Okay? Questions? And of course, to further aid in that, obviously, now here at the distal convolute tubules, you're taking these solids back. And the water that goes back is just passive, so it's not a massive amount of water. So then, as a last-ditch effort, since anti-diuretic hormone is already secreted by the stimulation of angiotensin 2, that anti-diuretic hormone is going to act at the distal convolute tubules, is whatever amount of water that is in here, it's going to suck all that back into the blood. So we say, screw the urine, man. You can make a drop if you, I don't care. We're going to take that fluid back so we can raise the blood volume so I can be happy again with the filtration. That's all it cares about, okay? But by caring about that, it actually has a very nice, profound effect on the plasma volume itself, okay? So if that is the case, let's say you're massively dehydrated, what happens to your urine output?
Decrease.
It decreases. And this is why when you guys are working on the floor in the critical care unit, one of the main things you need to always check for patient that has like an overnight stay, or patient recovered from surgery, or patient in the ICU that is under some kind of severe type of trauma or chronic stress, check the volume output. Because if the patient does not produce urine, that patient is about to die. If you don't produce urine over a 24-hour period, that is very, very bad. That means that your system, your blood, is massively volume depleted. And that's why the kidneys is just doing drastically as much as they can to take all that stuff back so that you don't lose that. Because if once that filtrate leaves the collecting duct, it becomes urine. And you cannot take urine back into your body aside from drinking it, which nobody wants to do, right? All right, make sense? Okay, so that's the reason why we have to hydrate these patients adequately. If the patient is going into the ICU for whatever reason, chronic stress, some kind of debilitating disorder, maybe some kind of, let's say, you know, they're in a coma or whatever the case is, you have to continue to replace their volume all the time and regulate that. Even heart failure patient needs some degree of volume deposition, but then you have to kind of have that nice little interplay between how much volume to give so that you don't overwhelm their heart and then cause them to kind of go into massive failure and stuff, right? So medicine's fun, guys. Just don't kill your patient, that's all. Okay, all right, now there's another thing that we need to add to this. There's a very important metabolic waste that is also being filtered and some degree secreted throughout this overall renal tubules, and that is called urea. You guys remember urea? Basically from metabolic breakdown of your amino acids in oxidative deamination and stuff like that. And then that ammonium that is released by that amine group basically gets taken into the liver for processing and becomes urea, right? So urea is released inside the filtrate and eventually it becomes urine. But if you are dehydrated or if you're volume depleted and the countercurrent multiplier system is now kicking up, to basically actively getting more water back into the body to conserve as much water as possible, then urea actually from this area gets transported out temporarily. Okay, so basically urea gets transported all the way into this space here between the descending and ascending nephron loop, and almost immediately it gets transported back into the distal convoluted tubule. But while it's in transit, while it's being transported throughout this, it's sort of like taking a scenic route instead of being deposited directly into the urine. For some reason, urea is like, you know what? We're gonna take you out of this and place you into this environment. And as you transition into this environment, you are now also contributing to the overall solute concentration inside this space. So urea helps to increase the osmolality of this space here so that you can draw even more water from the descending nephron. Okay, so urea aids the countercurrent multiply system by increasing the water reabsorption at the descending nephron because it adds to the overall osmolality of the space, so therefore the space now becomes even more hypertonic compared to the volume inside the descending nephron loop. Okay. This is going to reabsorb more water from the descending nephron loop, so therefore you can conserve more of that water. So that's the role of urea in this overall system as well. Any questions? I'm gonna erase this. Okay. Yes. I mean, you guys already have this. You should know it. You're telling me the story, so that's good. That's what I want to hear. Because I don't know it. You guys gotta teach me. I'm old, my memory's failing. So eventually, I'm gonna have to rely on you guys to teach me this stuff. All right. So now ask yourself this. Once all of that is said and done, right, the urine now, or the filtrate leaves the collecting duct, now becomes urine. The urine is collected into the minor calyxes, major calyxes, renal pelvis, and then down the ureters. As urine is propelled down the ureter, it gets deposited into the urinary bladder. Once it gets into the urinary bladder, you're gonna store that urine. Now, when urine is deposited into the urinary bladder, what do you think happens to that bladder? It gets distended, right? So, the bladder goes through a reflex called the mituration reflex. This is basically the sensation of pee or urination. Like, oh yeah, I gotta go bathroom, right? Because now my bladder's filling up with some stuff, who knows? I gotta evacuate all of that. Okay, so here's your bladder. The bladder has this dome shape on the top, and basically it just kind of comes down this way. Okay. Mucosal surface of the bladder is made up of epithelial cells we call transitional epithelium. And it has a lot of connective tissue and has a lot of smooth muscle. The smooth muscle basically is gonna fuse in together. There's like 3 layers of massive amount of smooth muscle that blends in together. So on the surface of the bladder, it forms what we call the detrusor muscles. Okay, so that's the detrusor muscle, which is a series of smooth muscle on the outer surface of the urinary bladder. The bladder narrows down into your urethra. Okay. Now, there's a portion of the detrusor muscle right here, what we call at the neck of the urinary bladder before it becomes the urethra. So there's a portion right here where the muscle actually thickens because all the muscle kind of condense together or converge together, right? And it thickens and it forms a sphincter. So the sphincter can regulate the passage or the flow of urine out of the urinary bladder. And we call this the internal urethral sphincter. Since the internal urethral sphincter is a continuation of your detrusor muscles of the smooth muscle, it is smooth muscle. And remember, smooth muscle is regulated autonomically, involuntarily, okay? Now, as the urethra is produced, it has to exit the pelvic diaphragm floor. The pelvic diaphragm floor is a series of skeletal muscle that forms the floor of your pelvic cavity, keeping all your pelvic organ in place, okay? So therefore, as it pierces through that pelvic diaphragm floor, it has to penetrate through a layer of skeletal muscle. So there's a layer of skeletal muscle here. Okay. And the skeletal muscle that basically wraps around that tubing of your urethra is referred to as your external urethral sphincter. And the external urethral sphincter is skeletal muscle. Okay, which can be voluntarily regulated. All right. Now, as urine is deposited into this particular bladder, it's going to stretch. So therefore, you have to have a reflex. Now, of course, any reflex requires sensory neuron and motor neurons to basically be part of that particular reflex, right? So you have to have a bunch of spinal cord segments here. Okay, 1 and 2. Okay, so this would be your lumbar spinal cord, and this would be your sacral spinal cord. Okay, be located somewhere in the lumbar. This would be somewhere in that L3, L4, or L2, L3 area, which is basically part of the sacral spinal cord. Okay, so for the micturition reflex, there's 2 different phases. One is called the guarding phase, and 2 is called the voiding phase. Alright, so for the guarding phase, as urine is deposited into this urinary bladder, so urine is deposited, and as urine deposits, it's going to start moving upward. So this is urine. Okay. Because the entrance of your ureters is in the back, and as it starts to fill the urinary bladder, it fills it from the bottom up. Okay. Now, if urine is deposited, and this is a very small amount of volume that is being deposited into your urinary bladder, it's going to induce stretching. And because the volume of the urine is not so much, the stretching is not that much, right? So we'll say low stress or low stretch, not stress. Okay, so low stretching. Now, this low stretching is going to be detected by sensory nerve. And the sensory nerve is going to relay this information to your lumbar spinal cord level, and also relay this information to your sacral spinal cord level. Okay? Because it's low stretch, you really don't want anything to be done. You don't want to initiate anything. So therefore, from the sacral spinal cord level, there's going to be an autonomic motor neuron that goes back to your detrusor muscle. This is known as your parasympathetic nerve. Okay. Parasympathetic nerve relays this information back and inhibits the detrusor muscles. Why? Because during the guarding phase, there's only a little amount of volume, so therefore, as the muscle stretch, it wants to contract, right? But since you're guarding against urination, against micturition, now the parasympathetic nerve will basically inhibits the detrusor muscle. So the detrusor muscles, even though it stretch a little bit, it's not gonna contract. Make sense? Okay. These nerves over here we'll call sensory nerves. Okay, that and this. Now, from the lumbar spinal cord level, there's another autonomic motor nerve, and this autonomic motor nerve is gonna go all the way here to your internal urethral sphincter. Okay, this is known as your sympathetic nerve. So from the lumbar spinal cord level, the sympathetic nerve is going to stimulate the internal urethral sphincter to contract. Does that make sense? You are guarding against urination. Okay, so therefore, the guarding phase is due to a very small amount of urine that is being deposited into your urinary bladder, causing this low stretch of the urinary bladder. So because there's low degree of stretching, the sensory nerve relay this information back from the sacrospinal cord level. It has an inhibition to your detrusor muscle via your parasympathetic nerve. So as the bladder stretches, now it inhibits the detrusor muscle from contracting, because you want to guard against that urination. From the lumbar spinal cord level, it stimulates the sympathetic nerve to now stimulate the contraction of your internal urethral sphincter. The smooth muscle contracts. So as it contracts, it closes off this passageway, therefore guarding against urination itself. Okay? Now what happens when you have more urine deposited? Because when this guarding phase is activated, you actually don't even know it's happening. Okay? Because it's sort of like a subconscious activity. So it's an autonomic response as far as the micturition reflex goes. But as the urine continues to be deposited, so now you have more urine. So because you have more urine, you will now have higher degree of stretch. Okay. Now this high stretch obviously has to be detected by your sensory neuron. So I'm going to draw this in blue. Okay, I'm just going to overlap it. It's the same thing. So imagine here, now you're detecting that. So this sensory nerve is going to relay this information back again to the lumbar spinal cord. And it's going to relay this information all the way back to the sacrospinal cord. Okay. From there, it's going to stimulate the parasympathetic nerve, which goes all the way back to your detrusor muscle. But in this instant, it's going to cause the detrusor muscle to contract. So in the voiding phase, because now you have more urine being deposited into your urinary bladder, it causes a more intense stretching. And because of that, the sensory neuron relays this information to the sacrospinal cord, stimulating the parasympathetic nerve. That parasympathetic nerve now goes back to the detrusor muscle and stimulate the contraction of the detrusor muscle. Massive smooth muscle contraction. Okay. At the same time, the sensory nerve relays this information to the sympathetic area, to the lumbar spinal cord. Now from here, it's going to stimulate a sympathetic nerve. And the sympathetic nerve, as it goes back to the internal urethral sphincter, is going to cause the internal urethral sphincter to relax. Make sense? So now you want to void, right? You want to get rid of this urine, so the detrusor muscle starts to massively contract, and your internal urethral sphincter starts to relax. So now urine is being pushed into your urethra. Now anytime you have this voiding phase going on, the sensory nerve that's relaying this information of that higher stretch will relay this information all the way up to your cortex. Okay. Unless you're a baby and wears diapers and have no sensation of urination whatsoever, we're all adults, we're like, you know what? I gotta go to the restroom because now my bladder is kind of stretched out, right? So we all have the feeling, that need to go to the restroom to evacuate our urine if we have more urine being deposited into our bladder, right? We all have that sensation. And why do we have that sensation? It's because there's a sensory pathway that goes to the cortex, goes to the brain, and stimulate the brain so that we can become aware. of that sensation, okay? So from there, the cortex says, all right, cool, thanks for letting me know. Oops. So once the cortex understands that, yes, there's a need to urinate, now you send this information all the way back to the sacrospinal cord. From the sacrospinal cord, it will now stimulate a somatic motor neuron. Because as you become aware of this sensation to urinate, to void, you now have a choice. So this is a somatic nerve. You now have a choice to either relax your external urethral sphincter in order for you to urinate, or because this lecture is so stimulating, you don't want to excuse yourself to go to the restroom, you're like, I'm gonna hold my urine in. But it's not okay for you to like pee in front of everybody. I mean, who knows, right? Maybe we're a small group, we accept it. If you want to pee, go for it, just clean it up afterwards, right? However, socially it's not really acceptable because we're adults and stuff like that. We need to be able to hold our pee or be able to get up and leave when we need to. So now you have a conscious effort to either contract the external urethral sphincter to prevent urination, or to relax it to allow for urination to occur. Okay, so you have a conscious effort to do either. All right. Have you guys ever in like a car ride and you play that game? You know the game? Well, maybe you guys don't play that game. We play that game. Anytime we see like an outer state plate, we have to drink a glass of water. And we'll pick a stretch of road when they say the next rest stop is 20 miles away or something like that. Of course, on the highway, you see out place states all the time. Arizona, Nevada, right? Every time we see one, we call it out and we drink water. So by the time we get to that rest stop, we're like just waddling to the bathroom because our bladder was so swollen up, right? So filled with urine. But of course, we can't be peeing in the car because that's just wrong. So therefore, we have to contract, like consciously contract the external urethral sphincter. Now, when the urine fills, continue to fill your urinary bladder, right? It fills from the bottom up. So all of this area on the side of the bladder is actually tightly bound to your pelvic cavity. But the dome of the urinary bladder is not. That's the free surface, which means as you continue to fill up urine, the dome of the urinary bladder starts to expand upward into the abdominal cavity, okay? Now imagine, and if you work in the ER, you may see some of this stuff. So imagine if you're sitting in the car and you have a full bladder, and we're talking about like excessively full, right? Of course, we're responsible. We have to wear seatbelts, okay? Whether you sit in the back or in the front, doesn't matter. In the back, you have a lap belt. In the front, you have that, whatever triangular belt that you have, so a shoulder belt and a lap belt. That lap belt happens to go right across the border between the pelvic cavity and the abdominal cavity. So therefore, if something happens, let's say you have a motor vehicle accident and you have a full bladder, what happens is the seatbelt tightens to prevent you from moving forward, right, through the windshield. But as it tightens, it increases the pressure into your abdominal cavity. And if it increases the pressure here, it causes this dome to pop. So you can actually rupture your urinary bladder if you have a full bladder and you have a seatbelt on. Okay? That's why when your parents tell you, go pee before we get into the car, they don't understand this. But at least they understand. They just don't want to stop for you guys to pee. But hey, by doing so, they're actually saving your life. Okay? Because what's the worst case scenario if you pop your urinary bladder? You die. You bleed to death. Okay? Actually, that's not the worst case scenario. The worst case scenario is if you have some kind of contamination or infection going on inside your urine and your urinary bladder pops, you may go into sepsis and you die an agonizing death, more so than just like losing blood altogether. Okay, so therefore, make sure you guys pee before you go home or get onto a car ride or whatever it is. Tell everybody to go pee and stuff like that. Saves a lot of time because you don't have to stop like 10 minutes and stuff. All right, any questions about the micturition reflex? Okay, so what's the stimulus? Yeah, it's a stretch, right? Due to the urine deposition, right? Sensory nerve relay that information. What's the integration center?
Sensory
Which one is the integration center?
oh,
Where's the integration of these information happening in? At the spinal cord, right? Either lumbar or sacrospinal cord. So at the synapses between the sensory neuron. And that could be interneurons in between too. I just didn't draw them there, right? Doesn't really matter, but there's a synapse. So the synapse itself is the integration center of that. And of course, you can see that for this pathway, there's also a sensory neuron that moves all the way up to the cortex. That's also part of the integration center. So therefore, from the integration center, you will have these different type of motor output, motor activities, either sympathetic, parasympathetic, or somatic motor. Okay? And the effector is the detrusor muscles, the internal urethral sphincter, or the external urethral sphincter. Depends on what it is that we're talking about. Okay? Now you guys have to relate to me whether it's the guarding versus voiding phase, which one is active and which one is inhibited during this time. It's not that hard, because guarding, you don't want to pee, so therefore you gotta do something to stop peeing. Voiding, you do need to pee in that time because you have more volume deposition, so you have to do something to allow for you to pee. But then consciously, you have the somewhat of a semblance of control to delay the peeing process until you get to an appropriate area for the evacuation of your urine. Good? All right, let's go to that.